Good morning. Today, let us start a new chapter, chapter 10, Microbes in Human Welfare. So, already we have learned in grade 11 regarding microbes in biological classification. So, we have learned about only different kinds of microbes and also some of the economic importance. But in this chapter, we are going to learn about what is the useful or the front part of the microorganisms. We know microorganisms four part also. Four means enemy. So microorganisms as a friend also, as a foe also. So in this chapter 99% we are going to learn about only the front part of the microorganisms. So microbes in human welfare, that is only we are going to learn. And microbes means you want to remember all microbes starting from Monera. Monera means prokaryotic group, bacteria. Okay, prokaryotes, Monera, bacteria. And second one, your fungi and some of the algae and protista group. Okay, protista we have learned so many protista group, chrysophytes, euglenoid, slime mold. Okay, so many protista, single celled eukaryotes also we are going to learn. So you want to remember first group Monera, prokaryotes, Monera, bacteria group, and protista group, single celled eukaryotes, and some of the fungi and some of the algae. This is only we are going to learn in this chapter in a detailed account. So what is our topic? Our topic is microbes in human welfare. Only as a friend, microbes as a friend we are going to learn, not a four part. Four part it will be in chapter 9, that will learn separately. So in this chapter, microorganism as a friend. So it is what is the human welfare part of the microbes, that is only we are going to learn. So we have discussed microbes are diverse. Diverse means they are diverse groups we can able to see. Different kinds of microbes will be there. Starting from protozoa. Protozoa comes under what group? Protista group, single celled eukaryotes. And bacteria, Monera, prokaryotes. Fungi, single, it is separate kingdom, according to R.H. Whittaker, it is a separate kingdom. And microscopic plant viruses. Okay, we have learned viruses also in grade 11. Viruses, viroids, and also prions, that is proteinaceous infectious agents. Okay, these are the diversity of microorganisms. So in what way it is going to help, only we are going to learn in this chapter, microbes in human welfare. So already we have learned about protozoa, that is your clear protista group. Okay, single celled eukaryotes. Bacteria, Monera, prokaryotes. Fungi, separate kingdom. And plant viruses, we have learned tropicomosaic viruses. Okay, so many plant viruses we have learned. And viroids. What is viroids? Viroids we can see only genetic material but no protein. No protein, only that we can call it as viroids, or naked virus only viroids. No protein, so only RNA will be there. That is only we'll call it as viroids. And prions. Prions, only protein will be, there won't be any genetic material. So how these kind of microorganisms will be helpful for human being only we are going to learn. Okay, so let us start. Microbes like bacteria and fungi can be grown in a nutrient medium to form colonies and can be seen in naked eyes. Now look at this structure, look at this Petri plate. This is only we can call it as Petri plate. Normally we used to grow microorganisms in a Petri plate in lab only. So for that we need to introduce our nutrient medium. Like see the nutrient medium like white color. This nutrient medium contains all glucose, protein, vitamins, minerals, antibiotics, everything this nutrient medium will contain. So this is very important for the growth of microorganisms. So this is only we are telling colonies. Now look at this, we can see here one colony, here one colony, here, like that. Colonies of microbes we can able to see through our naked eye. Individual microorganisms we can't see. For that only we are depending on microscope. So here we can able to see colonies of microorganisms. And how the colonies of microorganisms are growing means they need a nutrient medium. So when we provide a nutrient medium means, and also we should add a starter culture, and we can get our desired microorganisms in a Petri plate. Okay, and we have learned in the previous chapter biotechnology, after this Petri plate only we are taking, and when we are introducing into the bioreactor for huge production. Bioreactor for huge production. So this is growth of microbes in the Petri plate only. Okay, and some microbes causes disease and some are useful for human being, that already we know. Microorganism as a friend also, as a foe also. So friend part means will get more benefit, foe part means even human beings, plant and animals will get diseases. Okay children, so we are going to learn detail of different kinds of microorganisms, they are useful part. So first we want to know that all known matter only, anyway we'll discuss. So we know microorganisms will be ubiquitous. Ubiquitous means we can able to see everywhere. Microorganism can be seen in water, air, your body, dress, clothes and plants, animals, and also very hot temperature, also very chill temperature, also everywhere we can able to see the microbes. So we can tell it is an omnipresent or ubiquitous, both are synonym word only. For example, thermal vents of a geyser, okay, like temperature above not 1000 degrees Celsius, 100 NCRT is 100. So 100 degrees Celsius also we can able to see some archaebacteria, and deep in soil also, under snow also, we have learned in ecology, so wherever we can able to see, we can able to see in under snow also, and diverse nature we can able to see everywhere. These and all extreme means thermal vent we can see in extreme, and also deep in soil, under snow, this and all extreme climate. Apart from the extreme climate, normal climate we can able to see everywhere the microorganisms. And we know the category protozoa comes under protista, bacteria monera category, fungi separate kingdom, virus, viriod, prions, not in the RH Whittaker classification, but it is separate group, virus, virioids, prions. And these are the different kinds of microorganisms we can able to see everywhere. Not only everywhere, we can able to see in extreme climate like thermal vent, and also the deep in soil and also snow. What is thermal vent, children? So near to the volcanic areas, only we can see the thermal vent. So what is the useful of microorganisms? We can see so this is general concept. What is the useful of microorganisms? We can able to prepare antibiotics. And what is the harmful foe part of microorganisms? It is a disease. Okay, so here we want to see microbes. No, we are telling microbes as a front. Okay, we are not going to learn foe part, we are going to learn about front part. So under front part, in your NCERT syllabus, 6 heading was given. First one, microbes in household products. Second, microbes in industrial products. Third, microbes in sewage treatment. And fourth, microbes in production of biogases, and microbes as a biocontrol agents, and microbes as biofertilizers. So in these 6 headings only we are going to learn about different microbes. So first, under that first, microbes in household products. Already every day we are practicing without knowing that is a bacteria, or without knowing that we are doing some biotechnology procedure. Without knowing, every day we are practicing. Okay, first we'll see the microbes in household products. A common example in the production of curd from milk is the household products. Every day we used to practice, we are converting milk into curd by introducing a starter culture known as lactobacillus. Okay, so after 8 hours the milk was turned into curd. So here what is the role? The role is a microbe. So microbe as a front for human being. So we'll see little bit detail. So till lower classes we know lactobacillus will convert milk into curd. So how it will convert, that is only we are going to learn in little bit detail. So microorganisms such as lactobacillus. Lacto means why we are specifying lactobacillus means lacto means a sucrose, a sugar which is present in the milk is lactose sugar. Bacillus means a type of bacteria. It is a rod shaped bacteria. Now look at the structure. This is a rod shaped bacteria. Even we can able to see flagella. Flagella is there. So the rod shaped bacterium and it can able to change the sugar nature, okay, in the milk, lactose sugar. So that's why it is very specific, lactobacillus. And how we can commonly call means lactic acid bacteria, LAB. This word is very, very important. You underline in your book also, page number 181, second paragraph. Sorry, microbes in household product, under that fourth line. You underline please, lactic acid bacteria. Okay, this lactic acid bacteria only convert milk into curd, and it will grow in milk and it will convert it to curd. Okay. So how it will convert means, during growth the lab produces, lactic acid bacteria produces acids. What acid? Lactic acid. It started to produce lactic acid. So this lactic acids will coagulate and partially digest the milk protein, because now we need curd means we need curd protein, not a milk protein. So the milk protein should be digested by the enzymes. So here no enzymes or acids, but here acid was produced by lactic acid bacteria. Lactic acid was produced, so it will help to digest the proteins which is present in the milk. So while digestion it started to coagulate, the milk started to coagulate itself. For example, water means it is a plain solvent medium, and milk is also a solvent only. But when we add some chemicals means it started to coagulate, it started to become solid. That is only we can call it as coagulation. So here the nature of milk is going to change because of the adding of lactic acid bacteria as a starter culture for milk. So after adding, the lactic acid will started to digest the protein of milk and it started to coagulate, and we will get curd, and we will get a curd. For that we need a proper temperature. In that condition only this lactic acid bacteria will work, and lactic acid bacteria is an anaerobe. So we want to close and keep the culture. If we open and keep means we will get product, but we won't get that much effective product. And it is of they need a proper temperature. Without knowing, when we kept the milk after adding starter culture in the refrigerator, in the chill climate, it won't coagulate and digestion of protein won't be there. So for this conversion, a proper temperature and also anaerobic condition is must for conversion of milk protein into digestion of milk protein and also formation of curd. And formation of curd not only contains only protein, it contains very important vitamin B12. So what is the other name for vitamin B12? Cyano, cyano, cyanocobalamin, cobalamin, cobalamin, BALIMINE, okay, MINE, cyanocobalamin. You write and keep CYANO, cyanocobalamin, COBALIMINE, cyanocobalamin. Okay, and very, very important, curd is containing curd protein. Apart from that, it containing very important vitamin B12. Okay, what is the other name for vitamin B12? Cyanocobalamin. In our stomach too, the lactic acid bacteria play a very beneficial role in checking disease causing microbes. For that only we are insisting to take curd at least once in your day. So this lactic acid bacteria will kill the harmful bacterium which is present in your stomach and also your stomach part. Okay, so this lactic acid bacteria is very, very useful bacteria. Now this bacteria as lactic acid bacillus bacteria is available in tablet form also, because some people know the lactose sugar which is present in the curd, it won't suit for, or it will cause some allergy for the people. So those kind of people, they never take milk or milk product. So automatically they need a same kind of lactic acid bacillus to kill the harmful bacteria in their body. For that, supplementally we can take it as a tablet, like a tablet. So this tablet will create the condition to kill the harmful microorganisms in your stomach. Is that clear, children? So that's all about the lactic acid bacteria nature. So next video I'll continue the remaining. Thank you.